So I give the floor to Stefan Jansen, who is a microbiologist in Delft. Is our national lab for water research. So Stefan, is your session. So.
Yes. Thank you, Hadi, for the introduction. Well, I'm excited to facilitate the discussion about geochemical and microbiological effects upon hydrogen storage. It's a very important and interesting topic. As we already saw this morning, it came back in a couple of presentations. And it's not strange if you think that hydrogen is actually used as an energy carrier. And then it's not a strange question whether this energy can also be used in geochemical and microbiological reactions in the subsurface. And we are lucky today to have two experts here, one on microbiology, one on geochemistry, who can tell us a lot about these. And after their presentation, we have time also to answer your questions about it, and also to look out a bit further into the practical applicability of this. First speaker is Nicole Dopffel, who is already there. Nicole studied biology at the University of Tübingen in Germany, where she specialized in subsurface microbiology. And after that, she moved to BASF, where she was a research scientist. And from 2019 on, she moved to the Norwegian Research Center, NORCE, where she's a senior microbiological researcher. Next to the exciting research she does on hydrogen, a lot of you probably also know her as the driving force between a lot of initiatives which are focused on bringing the knowledge outside or together on hydrogen, such as the IEA TCP task 42. And also the Living on Hydrogen webinars that some of you might know. And I personally also know her as somebody who knows a lot of very fascinating details on microbiology and also who can tell very passionately about them. So I'm looking forward to the presentation.
Thank you very much, Stefan, for this very nice introduction. Yeah, thank you for the invitation, Hadi. It's great to be here again to just tell a little bit about the microbial aspects of hydrogen underground storage. Several speakers already touched upon it. Now I want to give you a little bit of information, some pictures. So we will start with a little bit of general knowledge on microbiology, and then we go into more details on hydrogen storage. So when we talk about microbes, the most of us have like these pictures in mind. Microscopy images of small unicellular round organisms. But I can tell you that these three pictures actually depict the three domains of life. So we have the three domains, which are the bacteria. Most of you heard of them. Then we have the archaea, which are not so well known. And then we have the eukaryotes. We belong to the eukaryotes. We are the animals, the fungi, the plants, and a few others. So these three domains of life, we've all come from a last universal common ancestor. So millions and millions of years ago, there was a unicellular organism, and then there was this major split in life. And then we had the bacteria evolving, and then we had the archaea evolving. And out of the archaea, then the eukaryotes came. The archaea are not so well known, and sometimes, but they are actually very important also for the subsurface. Bacteria and archaea are. And archaea are not like very strange, random microorganisms. Like recently I read an article where they were called like ancient types of microbes. It's not true, they're not ancient. You see, they have the same age as the bacteria. They are just not as well researched. And I think the main reason is because up until now, there are no archaea which are pathogens. So they don't cause any disease. And that's probably the reason why humans don't really care about them. So this tree of life is now, of course, very simple. You can imagine it looks a little bit more complicated when we look at the next. This is the most current tree of life based on the DNA information we have. So again, we have the three domains of life. And actually, the size and the distance of a line depicts the how different they are based on the DNA. And you can see the eukaryotes there we are actually quite uniform when it comes to our DNA. The archaea are also not as diverse. And then you see the bacteria. So they are extremely, extremely diverse. They're very, very different from each other. And you actually see also like every red dot depicts a major group which we are not able to culture in the lab. We only know of them based on their DNA we found. But of course, they are extremely important. So the bacteria and archaea, as I said, are the most important one when we now talk about the subsurface and hydrogen storage. As I said, they're both unicellular organisms, relatively small, and they can use a very diverse different energy sources. They have fundamental differences, as I said. I will not go into detail. My really goal is that you know that they exist, and they know they're different. And a very important aspect is also when you have CO2 and hydrogen in your subsurface, and there are specific types of bacteria, they will form acetate. They are the so-called acetogens. If you have special types of archaea present, you will get methane, the so-called methanogens. Up until this date, they have no bacteria ever discovered which can form methane or the other way around. That means when you have a reservoir and it's producing methane, microbial methane, you can already say to your colleagues, we have archaea. And then your colleagues will probably say, what? And then you can explain what I just explained. And it's really, I try to, archaea, they are there, they're important. And they're underestimated, I would say. I also, like yesterday, I read a text where they said, yeah, methanogenic bacteria called archaea. I can't even tell you how wrong this sentence is. This is how, like we would say, yeah, they are green animals called plants. And at least we would be in one domain of life still, so it's actually not that wrong as this previous sentence. So, but okay, moving on. So where do you find them? There's a very general rule of thumb that you can say in more extreme conditions you find more archaea, and in ambient conditions you find more bacteria. But it's really, there are plenty of exceptions, so it's really rough. And there are several factors shaping a community, and the most important factor is by far the temperature. As already mentioned before, we have a lifespan of minus 20 degrees to plus 122. This is what we know. There are living, growing microorganisms at these temperatures, as long as the water is liquid, of course. So there we have a really hard cutoff. And it's actually the only cutoff we know. When it comes to pH, all the pH ranges, you will find experts which can grow. Same with salinity. Salinity is no problem. You can go up to saturation, way beyond saturation. There are specialists which can grow. Same with metals. I mean, at a certain metal concentration, you know, a lot of copper, a lot of silver, mercury, that will, of course, at some point kill everything. Certain ions, then nutrients, what kind of nutrients are there, this is important. Permeability, when it comes to a reservoir, of course, so these cells, they need space. If it's a really, really tight reservoir, they just don't have the physical space to live. But this is rarely the case, I would say. And then also pressure. With pressure, we don't know too much the effects of it yet. But it could also be a factor shaping the community. And you may ask now, okay, but when we talk about the subsurface, how deep can we actually find life? And it's a very interesting question, and there were several research projects trying to answer that. Very famously, the one in the coast of Japan, where they actually drilled. They made a world record on scientific drilling, like several, I think almost 3,000 meters or 2,000 meters something into the subsurface, plus additionally 4,000 meters of water. So and receiving samples. So it's quite a achievement, I would say. And they found microbes up to 2,460 meters below sea floor, methanogens producing a little bit of methane from coal. And the cell numbers are actually extremely low, like maybe 10 per square cubic centimeters. They are really there, really active. Low cell numbers, but of course this subsurface, like this rock, it's enormous. So when you actually calculate now how many of these cells are there in the deep subsurface, you come to this staggering amount of cells. And this would actually be 12 to 20% of all the microbial biomass on our planet. So a huge amount of life. And so there's actually no depth limit found yet. It always coincides with the temperature. As soon as you cross the 122-degree line, it seems to be just a stop, and then it's sterile. Yeah, but it's fascinating when you consider this size of life. And I think sometimes it's a relaxing thought, you know, whatever we do at the surface, like, you know, war, climate change, an asteroid hits, these guys, they wouldn't care. They wouldn't even notice. They would just be there producing their methane once in a while, living their lives. And at least I think it's relaxing to think about it. It's a major part of the life on our planet, just there. Yeah, it's relaxing for them also, I guess. So what are now the metabolic activities of microbes? This is now a very typical soil, you can say. So you have your organic matter input, some kind of shape or form. It will be degraded into smaller organics, and then it can be fermented again by microbes into even smaller organics, also CO2 and hydrogen. Yes, microbes can also produce hydrogen, a lot actually. And then there comes now the different respiration types. So first of all, you will have oxygen. Oxygen gives a lot of energy. That's also the reason why we use it in our body. And that means also that your oxygen will be depleted very, very fast. So when you look at a typical soil profile, oxygen will be eaten up very, very fast. So put oxygen into the subsurface, it will be gone. And then there will come the nitrate reducers. They will use exactly the same things, but you're reducing nitrate. Then there will be manganese reducers, iron reducers. And then when that is happening, there will be sulfide reduction. Sulfide reduction, there you can find then the famous H2S. And if you still have a little bit of iron, you will get FeS. These are the black iron sulfide precipitates you always often see in microbial cultures, or when you pull something out of the subsurface, it's like these black minerals. And only when the sulfate is completely depleted, you will get the methanogens. This is a so-called redox cascade. That means they will compete, actually, all these metabolisms compete with the same electron donors and carbon sources. And this is also the reason why, for example, nitrate reduction delivers much more energy to microbes than sulfide reduction. So you will always have first, as long as you have nitrate present, you will not get sulfide reduction. First, when the nitrate is depleted, then you will get sulfide reduction, and when the sulfate is gone, then you will get the methane out of it. So this is very important to remember. So now when we think about hydrogen, hydrogen now is a very good electron donor for microbes. It has been around for a very long time on planet Earth. So all metabolisms are actually able to use hydrogen as electron donor. They actually can be fueled, as you can see, all of them can be fueled by hydrogen. So we are now trying to figure out what are actually now the most important factors when it comes to hydrogen storage. Is it the available electron acceptor? Is it the hydrogen concentration? Maybe other chemical characteristics? Is it the microbes present? Can we see something? But at the end, like the major sentence you can also remember is wherever you find living microbes, you should be concerned about your hydrogen. I think that is a fair sentence to say. So now with all this newfound knowledge you have, let's do a little test. When I say, whenever you find living microbes, so I will now show you some pictures with some basic information, and you will guess if you think there will be living microbes. So everyone in the room, I just say yes, and you raise your hand, and online you can put it in chat. Yeah, I will tell you the correct answer. So we start very simple that you get the hang of it. So our tongue. So 36 degrees, a little bit of salt, slightly alkaline. So who thinks there are microbes? And who thinks there are no microbes? Very good. Yes, of course, there are microbes, around 20 billion per human tongue. So a whole planet on each of our tongue. So next one. An active volcanic crater with lava around 1,250 degrees. Who thinks there are microbes? Who thinks there are no microbes? Okay, very good, yes. It's a little bit too hot. So it's, as I said, 122 degrees. Yes, yeah, I was really referring to the lava. And there are recent studies from Iceland. There was this volcanic eruption recently, and it was shown. It takes about a month when the lava cools down, and then it's completely occupied by microbes. Okay, you got it. Now it gets a little bit more complicated. Pitch Lake in Trinidad is an asphalt seepage. This is just pure asphalt. Freshwater salinity, 29 degrees. Who thinks there are microbes? Who thinks there are no microbes? Okay. So yeah, yes, you find microbes there, actually. They're really, really tiny water inclusions in the asphalt containing, actually, each water droplet contains a different community, and they are hydrocarbon degraders inside this pitch lake. So quite fascinating stuff. Rio Tinto in Spain, it's a river. It has a pH of around 2. Who thinks there are microbes? Who thinks there are no microbes? Yes, you find microbes. Actually, a lot of them. Yeah, it's very acidic, but there are specialists growing, and not even only bacteria, archaea, also algae. These are actually algae growing there. Okay, next one. The Black Pond in Ethiopia has like 800 grams per liter salt, 70 degrees, pH 3.5. Who thinks there are microbes? Who thinks there are no microbes? It's actually, there are no microbes. Yes? This is actually one of the very few examples of a watery body on our planet where you don't find microbes. It's just too much stress. It's high salt, high temperature, low pH. If you combine several stress factors on top of each other, it gets really, really tough for microbes to grow. So this is actually something where you don't get life. Okay, so the NASA clean room. Who thinks there are microbes? Who thinks there are no microbes? Yes, of course you find microbes there. NASA has a very, very strict, we should not contaminate our planet's policy, which I think is good. So they take very good care of their clean room, but still they are not able to get rid of them. They actually found a very specific one, Tersicoccus phoenicis. This type of microbe has only been found twice on a planet, in the NASA clean room and the ESA clean room. And it's extremely resistant against desiccation, radiation, biocides, solvents, low nutrients. It's actually, they recently discovered it is feeding on the cleaning products NASA is using. So they can't get rid of it. And you should remember this whenever somebody suggests you should sterilize your reservoir with a biocide. Then you can think of this one. You can't even get it off a clean surface. Okay, last one. Pure halite, 8.5 million years old. Who thinks there are microbes? Who thinks there are no microbes? You actually have microbes there. There are water inclusions in the salt. You see them under the microscope. And within these water inclusions, you find tiny little bit of microbes. inside the pure salt. Yeah, and now you can think about your hydrogen storage site. I think you now understand, like, even under really extreme conditions, you have very good chances that you find microbes. So when you have a salt cavern, it's high salt. But whatever, you know, but it's really the temperature which is really the key factor. So what can happen with a hydrogen storage and microbes? You can see here what actually are the suspected effects. You can have H2S production, methane or acetate production. You can have reactions with minerals, microbial influence corrosion, or increased growth and plugging. So we'll just show you now a little bit of data on some of these aspects. So first, H2S production. So here you can see now how it actually looks like when a microbe grows in hydrogen. This is a very well-known strain, Olei desulfovibrio alaskensis. You probably all heard of them. And like in dashed, it's the sterile controls of hydrogen, and then you have the different lines. It's just the different amounts of hydrogen, and you see as it feeds on the hydrogen. So it just consumes the hydrogen during growth. Now you would think, okay, now there will be a lot of H2S production because it's a sulfide reducer, it will produce H2S. If you now look at the H2S, it does very strange things. You know, it's like going up, down, up, what? And this is, in the first thing is like, it should just go up all the time. Like it's growing, so it should go up. So this is actually, you can explain it when you look at the reaction rate for sulfide reduction growing in hydrogen. And you see it's a proton-consuming process. So when you look at this graph again, you can actually see at the pH, we start with a pH 7.1, and we end up with the highest at pH 9.1. So this is because they are using hydrogen, they are using protons, the pH goes up. And as we know, H2S is very sensitive to pH. If you increase the pH, it will be in a dissolved phase. So you will not be able to actually measure it in the headspace anymore. So one major thing we now learned in the last year is that sulfide reduction on hydrogen will increase your pH. So always look out for it. I think it's a very good indicator. Now this is now a pure strain. I just grow in the lab. You will now ask, but is this actually relevant? So I have now, this is now data from my paper just published last week. So we took actually salt cavern brine, just brine from an old salt cavern, put hydrogen on top, no dilution, anything. And yeah, as you can see, like we started with a pH 7.5, and after 170 days, hydrogen was consumed, H2S was produced, and we end up with a pH of 8.5. So you actually can see H2S going up, H2S going down. It's because it's starting to be in its more dissolved form, because you push towards 9. So this already shows that a salt cavern brine has the potential, just at the salinity it has, it was 27% salinity saturated, that there are sulfide reducers. They can consume hydrogen and they will produce H2S. Also, if you look at the High Stories project, actually in the deliverable 3.3, this is now from MicroPro, Martin Wagner's group, you will find one graph, and it is the pH evolution. And as you can see, they start with a pH 7 and whoop, the pH is going towards 9. So I suspect this is actually exactly the same thing. They are growing on hydrogen, the pH is going up. You will actually have the same pH effect also with methanogens, because they're consuming protons and they're consuming bicarbonate. So now going to the next, methane production. So I want to show you actually one of the oldest microbial papers. It's from Smigan. They had this town gas storage in Lobodice. Everybody, I think, read about it in one kind of review paper. And they saw methanogenesis, as you can see, like methane production over time. So now, what are these different dots? I can tell you. So the lower part this is the original brine producing methane. then the other dots is the original powdered rock with distilled water. And it's still producing methane because the microbes are on the rock. And then the highest rate is you get it actually original powdered rock with original brine. So this already also shows that the original minerals you have in your porous media and your rock will have a huge influence, probably enhancing growth. So original minerals will have an effect. You will have cells attached, you will have nutrients, CO2 source, and you will have a buffering effect also. So whenever you can, you should include your original minerals. Now when it comes to acetate formation, this is now from a paper from Schwab. They also looked at acetate formation from an enrichment, different salinities. So the very light blue is the lowest salinity, so you have sulfate reduction very fast. Then the next line, sulfate reduction is getting slower, and then really high salinity. They got less sulfate reduction, but the acetate production always went up. So it's also like these acetogens, they're very, very resistant to a lot of stress. When sulfate reducers and methanogens can't cope anymore, there are very often acetogens taking over. And what we actually can say, we don't know too much about them. They're not very well studied, so there's a lot of questions about them when they are active and when they're not active. And then last point is about the increased growth. So this is now from my colleagues from the University of Bergen. You already saw some pictures. So we used a microfluidic device where we injected hydrogen together with the microbes, and you can see that the hydrogen bubbles get smaller and smaller and smaller, and they disappear. Of course, they have a huge surface area, these microbes, where they can attack the hydrogen. So this is also just showing that, like with a porous media, it gets quite easily consumed over time. But we also saw that at some point it just stopped. So it could be, again, we're not able to measure the pH because it was just too tiny, it was just a few microliters. But it could be, again, the pH actually being too high that they actually at some point just stopped consumption. And you saw this picture also, we actually observed wettability changes, so microbes can have a direct effect on that. So as also Hadi showed you, we have now done some experiments here at the TU Delft from Mate. I will not explain this because I can't really, but what we saw is that like using just a very rough sandstone, seeing the effects is very difficult. So we now had much more success with a polished surface, and we got beautiful SEM images of a biofilm affecting the quartz surface and then also affecting the contact angle. So also like on this really typical reservoir engineering aspects, wettability, also interfacial tension, and so on, dissolution probably of the gas microbes can have effect, either directly or indirectly. So, and with this I want to summarize. So what do we know for the salt caverns? So salt caverns are not sterile, I can say that. Until now, every salt cavern which has been looked at, there were microbes inside. And many of them are not well known. There are very, very high amounts of sulfate in these salt caverns, so the potential for H2S is there, and the activity will probably at the interface. It is in a closed system, so I guess the input of fresh cells, nutrients will be rather low. For porous structures, we can say it's very, very diverse. Every porous rock is different. Especially aquifers are very high in diversity. Up to 90 degrees, you can expect a lot of activity. And porous media gives you a large surface area, a lot of contact points for biofilms. Minerals can buffer, can deliver nutrients. And we also know from the oil and gas, as soon as you have a very active reservoir, it's almost impossible to control it. If you start microbial souring in a porous reservoir, it's getting tough to control. So what do we need to know? These are now the questions. So like with the caverns, where do they come from? I think that is a fascinating question. Probably not so fascinating for operators, but I think it's a very nice thing to look at. How long can they actually uphold this activity? Will this pH effect actually at some point self-limit them? That they just increase the pH so they can't survive anymore? How much H2S will be formed? How much will be in the gaseous phase? Will the pH increase, as I said? What mitigations options do we have in these salt caverns? It's a tough for chemistry, that's for sure. In the porous reservoir, we have many more questions, like which reservoir are suited. I guess high temperature, high salinity in combination will be quite good to look at. Then again, how long can they uphold the hydrogen consumption? Are there limits in nutrients? What are the interactions with the minerals? Will the biofilms, can they plug the pores? What kind of effect you will have on actually imbibition and drainage? And is there any way of inhibiting or mitigating this microbial consumption in a porous media? Or can we do anything about it? Because it will be very, very difficult, especially at large scale, to influence microbial activity. Yes, and with this I want to thank a lot of people I work with, especially my colleague Annie. And I want actually to thank everyone who's contributing on microbial aspects, like the University of Wageningen, Stefan and Jan at Delft, a lot of MicroPro. So it's everything we do is actually really helping us to understand this problem a little bit better. Oh, thank you.
Well, thank you, Nicole. Again, a very fascinating talk, a lot of interesting details, fun facts, even. Yeah. So interesting to hear. I'm sure there will be some questions. So, are there any questions? Yeah. And now there's time for a couple of questions, and then there will be another. Exactly. Yeah. Yeah.
Mm.
In some salt caverns, which are being used for gas storage at the moment, the sun, which is the mostly sulfur materials actually sticking out of the water table. Would you care to speculate what that would do to the microbial system, and are studies like this, is that configuration being studied?
Yeah, I can tell you there are no studies on this. I mean, there are only a few studies on the sump itself because it's so difficult to get samples. And if there are studies, they're also very rarely published, which I always like when there was like which things we should not focus on, and there was like papers, no, please publish, please. We need more knowledge. So the thing is, like the microbes, they need water, that's for sure. But as you saw with the asphalt lake, they don't need much water. But of course, the more water you have, the more cells, the more activity. But until now, we don't even know if there will be like highest activity at the top of the sump or the bottom of the sump. There's actually also, I mean, these caverns are not completely also dry. There will be a water film on the walls. Exactly, there is actually rain in a cavern when you have these pressure changes. So we don't know anything what microbes actually do there. So can't tell you. We need to investigate, yeah.
There is a online question is about if we cannot really avoid this, then what's the remedy if we accept that there will be microbial activities?
Yeah, good question. It depends now when you talk about like salt caverns versus porous media. I think with the salt caverns, because they're a closed system, I think we have good chances to get this working somehow. With the porous media, if you have like low temperature fields and everything else, getting this under control, I don't know, like with, as I said, like mitigation options, inhibition options, specifically for hydrogen consumption have not been done yet, or at least they are underway and have not been published. So,
No antibiotics.
no, that's for sure.
That's